The surprising truth about modern gene editing is that targeting the DNA is not the hard part. The hard part is what the cell does after you hit the target. If you do not control that repair step, you do not control the product. As of 2026, CRISPR-Cas systems are the dominant programmable editing platform across research, agriculture, and therapeutic pipelines. The toolkit is bigger than the original cut-and-paste idea. You have base editing, which can change a letter without a full cut. You have prime editing, which can write a small intended change more directly. You also have epigenetic modulation, where you tune gene activity without changing the sequence, and you can target multiple sites at once, which matters for traits that are not single gene stories. But performance is gated by post-targeting biology. After a cut, or even a nick, cells choose repair pathways. Non-homologous end joining is fast and often messy. Homology-directed repair can be precise, but it is harder to invoke reliably. Microhomology-mediated end joining adds another pattern of outcomes. Local sequence context shapes what happens too. So guide design is necessary, but it is not sufficient. In practice, editing is a probability distribution, not a single deterministic operation. That leads to the first strategic takeaway. The rate-limiting step is outcome control, not editing chemistry novelty. If you cannot bias repair toward what you want, you will burn time on rework. You will also struggle to scale, because the variance becomes the cost center. The second big finding is that verification is becoming the real bottleneck and the real moat. Off-target assessment is moving toward whole genome sequencing of edited organisms. That shift matters because it changes the argument from "we think it is clean" to "we measured the whole genome." A concrete milestone. In 2021, whole genome sequencing was used to analyze off-target mutations in CRISPR-edited rice plants. That work demonstrates what the validation stack looks like when you treat the entire genome as the audit surface. Even with whole genome sequencing, residual risk remains. Low-frequency events can hide in sampling. Structural variants can be harder to interpret than simple single-letter changes. Repetitive regions can be difficult to map, so verification is not a box you check once. It is a capability you build with throughput, depth, analysis workflows, and reporting that can survive dispute. A third finding is that agriculture is the highest volume deployment domain for gene editing right now. That is where you see repeated, real-world use cases. Disease resistance traits, climate-smart traits, ornamental traits, and general crop improvement are already active areas. Rice shows up often as a focus for disease resistance strategies. Cocoa is another concrete example, with cocoa genome engineering using CRISPR-Cas9 documented in 2022. When you want edited traits to become bankable, agriculture forces the issue because the field is the truth machine. Field truth also introduces a different kind of validation requirement. A trait that looks strong in controlled conditions can degrade across environments, seasons, and generations. Durability under stress is part of the product definition. That is why multi-environment trials and long-run monitoring are not optional accessories. They are what turns an edited genotype into an economic asset that capital allocators can underwrite. Now, zoom out to the constraints that surprise technical leaders the most. Governance and social acceptability act like engineering constraints. Ethical, legal, and social issues are not just commentary after the fact. They determine what can be deployed, where, and at what pace. Fragmented governance can create regulatory havens, which then undermine trust everywhere else. A single high-profile failure can shrink the deployment envelope for years. That connects directly to risk. Off-target mutations can create unintended phenotypes, including long-latency harm that is hard to detect early. Ecological disruption is a concern when edits affect fitness in open environments, especially with gene drives. Dual-use risk rises as editing capabilities diffuse and become cheaper. In this domain, the technical and the institutional are inseparable. So what should a senior strategy or capital allocation leader do with all this? Start by funding the unglamorous parts. Build repair and outcome control tooling that reduces variance. Build genome-wide validation capacity, including sequencing and interpretation, sized for real throughput. Build field validation networks for agriculture, because that is where durability data is generated. Treat governance integration as enabling infrastructure, not compliance overhead.